Let us move on to the next one, ice recrystallization inhibition. So far we had seen about the thermal hysteresis. Now we will move on to the next mechanism, which is ice recrystallization inhibition proteins. So the second type of antifreeze activity exhibited by the biological antifreeze is their ability to inhibit ice recrystallization. So inhibiting the ice recrystallization results in small ice crystal within a frozen sample. So one is called isomass recrystallization. This mainly involves internal changes within the individual crystals, reducing their defects and creating smoother rounded surfaces. So rather than the crystal, they form like a globular type materials. The second one is accretive recrystallization. This refers to the merging of adjacent crystals, reducing the number of boundaries and thus the total boundary area. The third one is migratory recrystallization, or called Oswald ripening. It is the growth of larger crystal at the expense of the smaller ones, driven by difference in curvature and surface energy. Let us look at these three types. So what is the definition? When we look at the isomass recrystallization, there is a change in the shape or internal structure of the individual ice crystals. Irregular grain surface become rounded and defects are reduced. And when we look at accretive recrystallization, two or more neighboring ice crystals merge onto a larger one. And when we talk about the Oswald ripening, large ice crystals grow at the expense of the smaller ones. This occurs at constant ice volume fraction, temperature and pressure. So here are the different process types are given. And when we look at the thermodynamic driving force for the ice mass recrystallization, it reduces the grain boundary area by rounding the crystal surface, lowering system's free energy. When we look at accretive recrystallization, this reduces the grain boundary area as the crystals merge, decreasing the free energy. And when we look at the Oswald ripening, this reduces the total surface area by eliminating smaller crystals, thereby lowering the free energy of the system. So what are the key characteristics? Those are given here. And what is the effect of crystal size? There is no net change in the overall crystal size, just a change in shape with respect to isomass. That's the reason we say isomass. The mass remains the same. And when we talk about the accretive recrystallization, the crystal size increases as the neighboring crystals merge. And in the Oswald ripening, overall increase in the average crystal size as smaller crystals disappear. And when we look at some of the examples, in the case of isomass recrystallization, minor reorganization of the ice crystals such as the ice under pressure happens. And in the accretive recrystallization, coalescence in ice-rich environments like ice formation in glaciers happens. And Oswald ripening generally happens in the ice growth in cold storage or biological systems where the large ice crystals are determinant. And here are some of the other things like how the role of the ice binding proteins affect these three recrystallization process. The IBPs basically inhibit the process of isomass recrystallization by binding to ice crystal surface and preventing shape changes. In the case of accretive recrystallization, the IBPs inhibit the process by preventing crystal coalescence through surface binding. And when we talk about the Oswald ripening, IBP inhibit growth of large crystals at the expense of smaller ones, effectively halting the process of Oswald ripening. And let us look at the comparison between thermal hysteresis and IRI, that is the ice recrystallization inhibition. So the basic definition we already know. The thermal hysteresis basically lowers the freezing point, thereby creating a gap between melting and freezing point. Whereas in the ice recrystallization inhibition, it prevents the growth and merging of ice crystals during freeze-thaw cycles only. And the mechanisms are given here for the thermal hysteresis. It inhibits the unlimited growth of ice crystals in body fluids. Whereas in the case of ice recrystallization inhibition, it maintains small non-lethal ice crystals to avoid cell damage. So in one case, the ice formation or the growth is inhibited. Another one, ice in small sizes are maintained. They are also not permitted to grow to a bigger one. When the thermal hysteresis was first discovered, it was first described by De Vries in 1969 in polar fish. And IRI mainly focuses on protecting cell from freeze-thaw stress only. It was not known when it was actually first reported. And other things are given mainly if you look at the application. The thermal hysteresis is common in psychrophilic organisms like fish and insects, whereas the ice recrystallization inhibition is essential for organisms experiencing fluctuating sub-zero temperature. So we will be actually looking at freeze avoidance and freeze tolerance. There, these two activities are very important. So let us look at what is the difference between freezing avoidance and freezing tolerance. Freezing avoidance means supercooling body fluids to prevent ice formation, whereas freezing tolerance means it is the controlled ice formation in extracellular spaces, because ice, basically these many of the marine mammals, animals, they live in the cold water. So they have to have, the ice will still be there outside, but they have to control the ice growth formation. So especially in the extracellular spaces and outside also, they have to be very careful. So freezing tolerance is being carried out by some of the fishes. So what is the key mechanism by which these two operate? The freezing avoidance depresses the freezing point of internal fluids, and in the freezing tolerance, this allows some extracellular ice formation while avoiding intracellular freezing. So this is what is the major difference. And the effective temperature, if you look at freezing avoidance, means it is effective in extreme constant cold, that is typically below 5 degrees centigrade, whereas freezing tolerance tolerate temperature from 5 degree to minus 40 degrees. So this has a very large range of tolerance. And when we look at some of the examples, the freezing avoidance is found in areas with constant extreme temperatures, whereas freezing tolerance species are found frequently in organisms inhabiting variable cold environment. And what is the cellular impact in the case of freezing avoidance? It prevents ice crystal formation in cells, keeping water in the liquid form. Whereas when we talk about the freezing tolerance, 2/3 of the frozen body liquid fluids considered the limit of tolerance. We'll move on to the next one, which is the ice nucleation. So ice nucleation is also another mechanism by which the ice crystal grows. There is an energy barrier which is called the nucleation barrier that must be overcome to form a small critical nucleus of the ice in a supercooled aqueous solution. This nucleation barrier prevents spontaneous ice formation in a supercooled liquid below its equilibrium freezing point. Let us look at the supercooling. Supercooling inhibits ice formation within the tissue. The liquid remains in metastable state until the supercooling point, that is, or the nucleation temperature is reached. This keeps the water in the liquid state. At this point, the liquid becomes unstable and the ice-like clusters start to form, growing into fully developed ice crystals. So the cell avoids extracellular ice formation. So what is the role of the ice nucleating proteins? These proteins play a critical role in limiting supercooling and triggering the ice nucleation at elevated sub-zero temperatures. These proteins provide a surface that facilitates the formation of ice by templating the organization of water molecule into an ice-like lattice structures. So what is the mechanism by which this operates? The ice binding surface of the ice nucleating proteins interact with water molecules, encouraging the formation of an organized ice-like lattice structures, which lowers the energy required for nucleation and promotes ice formation. These INPs effectively lower the nucleation barrier, enabling the ice formation process to begin at higher temperature compared to the pure water. So this actually happens in the extracellular regions. What is the biological role of INP? INPs are found in various organisms and help these organisms survive in cold environment by facilitating ice formation in a controlled ways, such as preventing freezing damage or aiding in cryoprotection. So here is the big chart talking about the ice binding proteins. We have antifreeze proteins and ice nucleating proteins. In the case of antifreeze proteins, the main two activities are thermal hysteresis activity and ice recrystallization inhibition activity. So when the TH is more than IRI, which is actually seen in few fishes and insects, when TH and IRI are more or less equal is seen in some of the bacteria, yeast, fungi and algae. And in the case of plants, the thermal hysteresis is lower than IRI. So that is how these proteins actually function in different types of species. And when we talk about ICE nucleating protein, this is mainly found in gram-negative pathogenic bacteria. So here is an example of how the ice crystal growth is being controlled by the AFPs. So here AFP3 is actually present here. So this actually prevents the growth from the A axis, and that's the reason the growth actually happens only in the C axis direction. And here are the basal planes that are given shown here, and this is the C axis through which the ice crystal growth actually happens.